Hello, my name is Brian Ratnasinghe, and I work with the Mello Center for Genomic Sciences and Precision Medicine, which is a part of the Medical College of Wisconsin. And here is my poster for ISCB. So let's go ahead and just get right into it. Don't have much time, so we gotta get moving. Uh, this poster is all about KRAS and MD enhanced sampling of KRAS. Basically the big idea here is that we want to be able to take a look at, a way to distinguish between different hotspot variants, of KRAS just using MD. And what we think is that enhanced sampling is a good way to do that, specifically Gaussian enhanced sampling, uh, you know, specifically on a lower time scale. I think it could be very useful for scoring, and then maybe being able to, in the development of this kind of scoring workflow for KRAS, we might be able to streamline the process, moving forward down the line with other proteins as well. KRAS, of course, great target for this since it's such a great example of protein that has a lot of data on it. And there's a lot of different information, a lot of research trying to assess all these different types of hotspot variants. So we have a lot of information on it. For anyone who doesn't know, of course, KRAS is a very vital protein that controls cell proliferation and growth. And so, the idea is that because of that, all these different, cancer variants, they're cancerous because they're changing the amount of cell growth and proliferation, right? We're talking about overgrowth of these of tumors, right? So let's go ahead and move into some of the data here. What an important feature of KRAS is the switch 1 and switch 2 region. That's kind of where the conformational change is going to be, and that's where KRAS actually controls different interactions with varying proteins, including the nucleotide exchange. And so in MD, of course, we do see conformational change in switch 1 and switch 2, which is different, between our wild type and the different variants that we're simulating. Again, the variants that we're taking a look at here are G12D, G12C, G12V, and G12R. And so let's go ahead and move forward. So basically, we took a look at comparing between switch loop conformations between the MDs. We've also been comparing between experimental structures. And so here is actually coordinate PCA, of the actual MD structures, and overlaid on top of the coordinate PCA, in varying colors are the different X-ray structures. Now, KRAS, of course, again, well studied. There's 200 different X-ray structures that I've now taken coordinate PCA of and overlaid on the sampling space of the different MD conformations. And what we can see here is that using Gaussian enhanced sampling, the X-ray conformations under 40 nanosecond simulations overlays quite nicely over the MD sampling spaces comparatively to unbiased, and there's a lot less overlap in space between X-ray and the unbiased. So that's something interesting. We at least determined that they could be quite reasonable to use Gaussian enhanced sampling, at least at this lower time scale. Moving forward, we decided to take a look at larger timescales. Here we took a look at some 2 microsecond simulations from a PLOS CV paper and compared them to 120 nanoseconds in Gaussian enhanced simulations. And we found that 2 microseconds, it can't be understated, that's a lot longer than 120 nanoseconds. Yet, of course, there's more space being sampled within the unbiased, no doubt about that. But we're sampling a reasonable amount of space, comparatively speaking, qualitatively, within our Gaussian simulations. And so we were trying to see, is Gaussian good enough, to actually be, using that, on these smaller timescales, 40 nanoseconds to 120 nanoseconds, for differentiating between the variants. We might not be sampling everything, but we might be able to differentiate on a strong enough level, to be able to score properly. It's just an idea. So of course, one thing that we've been playing with is the idea of being able to score the PC coordinate spaces. So here we've taken PC1, PC2, PC3, for all the different variants and compared them to wild type. And so J12C for PC1, for instance, is plus 1.6 standard deviations away from wild type in terms of PC space. And we get to know exactly what that means thanks to MD. Here's a rendering of what the different PCs mean. So PC1 is a very lateral movement, of switch 1 as well as switch 2. And then PC2 actually represents, switch 1 moving kind of downwards instead of away from the core of the protein, whereas, switch 2 here actually kind of has a weird, interesting twisting motion. And so it's interesting. So we get to know, what kind of conformational change these PCs mean, get to categorize, what all of the variants mean in terms of switch 2 and switch 1 different types of conformation, and compare between all these different sampling types and all these different types of variants. It's very powerful, and we think this is going to help us, down the line in a lot of different ways. Moving forward, we're going to keep on categorizing, keep on seeing what these mean, and then start moving on different proteins. But then also, take a look here. So the switch 1 is, very important for characterizing nucleotide exchange factors, right? And the ability of KRAS is to actually bind to these nucleotide exchange factors, right? And so you can see that in the bottom right here, different conformations of that. And so moving forward, we're going to take a look at maybe, what are these PCs relative to these nucleotide exchange factor proteins, right? There's a lot more to talk about, but unfortunately we're out of time here. Please feel free to, if you're going to be in person, come up to my poster and say hi, and we can talk more about this, of course. If you have any questions, feel free to DM me on LinkedIn. Let's see, you can find my LinkedIn. Well, my name is Ratnasinghe, and you can find my LinkedIn. Let me just go ahead and, oh yeah, there we go. Here, yeah, so go ahead, linkedin.com/in/Brian-Ratnasinghe. Shoot me a DM, we can talk more about KRAS and enhanced sampling. I'd love to. All right, well, have a great day. Hope you enjoyed this, and see you around.